So, good afternoon, everybody. Today we have Professor Francois Joanny from the Collège de France in Paris. He's going to present a talk on the physics of biological tissues. Professor Joanny made his thesis under the direction of Pierre de Gilles de Jeune, if I understand well. And then he moved to California for a postdoc. Then he returned to France and was professor in several universities in Lyon, in Paris, also at the Institut Curie. And later he was director of the ESPCI, the École Supérieure de Physique Chimie Industrielle de Paris. And after that, he became professor at the Collège de France for his many contributions in soft matter, from colloids, droplets, polymers, and now in this active subject of biophysics and biological tissues. So I asked Professor Joanny to make a general talk on the physics behind the description of biology, or biological tissues, to be more precise. So please, Jean-Francois.
Okay. Let me. It doesn't want to go full screen. Okay. I'll open it again.
It just worked.
It worked earlier, it doesn't anymore. I know. It's behind. Okay, so thank you very much for the introduction. Thank you also for the invitation. As I was saying, this is the first time I give a talk at this time of the day for me. And I'm very happy to give a talk in Santiago de Chile, which is pretty far from where I am. What I want to do is talk with you about tissues, so tissues in our body. This might be kind of unusual for physicists to work on such subjects. What I will do is give you a general overview of what we've been doing, and I'll try to show you that there is very nice and interesting physics and very various physics to make on this kind of systems. Before I start, I just want to set the stage and show you the types of tissues that we are working on. These are what are called multicellular spheroids. They are cell aggregates. They are made of cancerous cells. You see on all these images, you see the cells one by one in these cell aggregates. The size is of the order slightly less than a millimeter. And these are used by people to test drugs against cancer. So they are made with cancer cells and people use them to test drugs. This one here is a textbook picture of the inner surface of the intestine. You see here that there are protrusions going into the intestine. They are called villi. And in the middle of the villi here, you have things called crypt. The villi are extremely well ordered. They make a square lattice here. And what I want to show you is what happens if you cut the villi perpendicular. So you see that. And there's a layer of cell that lines the surface of the villus. And this layer of cell is called an epithelium layer. And I will show you a few things about epithelial layers. The last example is more of a physicist approach to tissues. These are cells, cell lines, which are plated on a substrate. There are monolayers of cells. So this one is seen from the side, the green color, and you see the cells one by one. And if you look from the top, you see the borders, the frontiers between cells that appear from here. So this is this kind of things that I want to discuss. In fact, there are kind of two ways to look at tissues. Either you can say, I will look at the tissue at the scale of the cell and how the cells are arranged between one another. And the model that people use for that, especially for these epithelial tissues that I was talking about, are called vertex models. And they are inspired from what people do from foams, in particular two-dimensional foams. I will be very brief on that, but I wanted to mention it. And then there's another way to look at tissues, which is kind of to consider them as materials, which means to use continuum mechanics. Of course, one should be able to bridge between the two approaches. It's not so easy, and I don't know a very clean or systematic method to do that. So after presenting the cell level, I will present what I will call a macroscopic or hydrodynamic theory. And I will give you two examples, and examples where the tissue or the spheroids that I just saw behave as liquids, and an example where they behave as solids. In the last part, I want to show you that considering the tissues as simple liquids in certain cases is not enough. And I will try to show you that we should consider them as active materials or active fluids, and in particular, nematic active fluids. So let me start with these epithelial tissues. So this is another image of epithelial tissue. This is the wing disc of the fruit fly Drosophila. This is a region of the pupa of the Drosophila from which the wing grows. And this is different stages. This is this monolayer of cells. It's seen from above, from what's called the apical side. And you see this network, which is the frontiers between cells. Now, of course, the cells are three-dimensional. So if I look at them in 3 dimensions, I just drew them in this very ordered array like you have here. And they are kind of cylinders which are confluent or packed with respect to one another. What people do to study these tissues, they only care about the surface of the tissue. So I will only care about this picture here and try to infer from this picture what happens to this monolayer of cells. And there are ways to justify that I don't want so much to go into. The theory starts from the fact that this looks very much like a two-dimensional phone. So what people do is that they point out all these junctions between cells here, these points, which are the red points here. And so these are vertices, and they put edges between the vertices, and they impose the fact that the edges are straight line. So if you do that, you get a representation, for instance, of this quasi-hexagonal thing as some kind of paving of the plane with edges, vertices, and cells. So this is how I want to describe these monolayers of cells. So the way you want to describe them is to write a force balance at the level of each cell. And a very efficient way to write a force balance is to write what I will call an effective energy function. There is no thermodynamics for tissues. They are living materials. They are fully out of equilibrium. Still, you can derive force balance from some effective energy using virtual work theorem, for instance. And here is the type of energy that people use. It was first written for foams by Francois Joanny in Grenoble then, and it was extremely developed by Francois Joanny in Dresden. So this energy has 3 terms. For each cell, we will assume that there is a preferred area, which I call A0, and all the cells have the same preferred area here. And the area tends to go to this preferred area, and there's a stiffness that I call A, which measures the energy cost of being different from this preferred area. So that's the first term. The second term is that on all of these edges, there is a tension, which is a line tension. So this is just a line tension term. This is a sum over all the edges of the paving, and the line tension is lambda. And there is a third term, which is proportional to the square of the perimeter of each cell. This term is here. Not everybody is using this term, but this term is kind of here to take into account the active effect of the cell which tend to contract on themselves. So this is what I will use. And there are a few things which are easy to do with this energy. The first thing is that people looked in great details in the book of Francois Joanny at the ground state. And I made here some kind of phase diagram for the ground state. If you make everything dimensionless, in effect, there are only 2 parameters. There is gamma, which is what's called the perimeter elasticity here. And there is lambda, which is an effective line tension. And all the other parameters are absorbed by rescaling. And there are essentially 2 ground states for the tissues. If lambda is positive or large, at least above this line here. The ground state is a hexagonal lattice, although lambda shouldn't be too large. If lambda is too large, the tissue is unstable. So this is this ordered state here, and you show one in the previous slide. If you are below this line, the minimum of energy is the absolute minimum of this energy, 0 or a constant, but it's very degenerate. So there are many states that have the same energy, and there are soft modes that correspond to jumps between these states of the same given energy. Now, of course, the tissue doesn't explore only this ground state. It doesn't explore only this ground state because it's evolved with time. And the evolution with time is due to what people call topological transitions. One of them is very simple. It's cell division. So when a cell divides, the naive stuff that you can do is you cut the cell into here and you replace it by 2 cells. And that then you let the system locally re-equilibrate. As you introduce cell division, you can introduce cell death. In cell death, you eliminate 1 cell. And again, you let the tissue re-equilibrate. And there is a third one, which in foam physics has been called T1 processes that correspond to an exchange of neighbor. I put a small sketch here. You see that these 2 hexagons are neighbors here. If you shrink this link, you get a 4-point vertex, which is unstable, and it can separate in the other direction. And after this T1 process, these 2 cells that were neighbors are not neighbors anymore. And these 2 cells that weren't neighbors become neighbors. So introducing these 3 types of topological processes, you can see how the tissue evolves. Now, what I will do is show you a movie that comes from the group of Francois Joanny. So you see the cells growing, and there is a color code. The color codes correspond to the number of sides of each cell. Now, what can you do with such a theory? The first thing that you can do is you can compare the results of such a theory with biological data, and people did that a lot for the development of Drosophila, which is kind of an example for development of animals. You can also try to develop the theory. I was involved in kind of 3 things. One is looking at the stability of this vertex, this ground state with respect to perturbations. And I did that with Rodrigo and Fernanda Perez. We looked at 2 perturbations, one that we call active, which was a change in this reference area A0, and the other one is a pre-stress imposed on the tissue. The second thing, which I think is important and which is kind of developing now, and which we did with Edouard Anzo, was to take into account the 3rd dimension. So the idea is very simple, just to go back here and look, for instance, at the periodic structure of the tissue, but taking into account the 3rd dimension. And in some cases, if you do that, you can show that the 2-dimensional theory is sufficient and reduces exactly to that. The last thing that we are trying to do now with a postdoc called Doron Grossman is to coarse-grain this vertex model to get a continuum theory for the tissue in 2 or 3 dimensions. This is not so obvious and so easy, so I won't talk about this here. What I will do is I will start from the other end, and I will show you how to describe tissue using what I will call something like continuum mechanics. So what I want to do that with is what I will call cellular aggregates, which are aggregates of cells. I will show you a few of these aggregates here. One type is these spheroids that I already showed you. So this one is a spheroid made with cancer cells, but other cells make spheroids. These are spheroids made with neural cells, progenitors of neurons. They aggregate and they make these spheroids that people call neurospheres, and they essentially have the same properties as the cancer cell spheroids. If I go back to this one, so the spheroid is here, but it's inside what people call a capsule, which is a thin layer of polymer, which is around here, which is a thin elastic layer. The idea is that when the spheroid grows, it will touch this elastic layer around. It will push on the elastic layer, which is a soft solid, it's a rubber, so it will stretch. and that exerts a control force on the spheroid. Now, what happens when you do there's a movie behind, I can send it. What happens, so here it doesn't touch, and it grows, and you will see that at some point. Here it starts to touch the external layer, it still grows, there is a force acting on it, and so it grows because cells divide, and when you apply a force, the cell division rate decreases pretty much, and at some point it stops to divide, and you reach a steady state, and the steady state is a balance between the force exerted by the external capsule and the tendency of the spheroid to grow. I have a last type of aggregate here. I mentioned the intestine in the beginning. This is kind of a people call it a microgut. It's called an organoid. It's made starting from one cell, which is a stem cell. If you put it in the right condition, in solution, it grows, and it makes this organ here. So it's kind of a small sphere, and you see protrusion going out. I told you that there were villi and crypts in the intestine, and this has all the properties of villi and crypt. So this is now considered as a much better model than the spheroid to study organs, and in particular to study drugs against cancer in these organs. So I won't talk about this once. I will come back to these capsules in a minute. Before that, I want to show you some properties of these spheroids which are here. So this is a picture now of two spheroids. The first thing which is obvious is that they are spherical, and if you're like me, used to things like capillarity and things like this, the idea that you can have is that this is like a fluid with a surface tension. And indeed, it's pretty much like a fluid with a surface tension. The idea of the surface tension is like the original idea of surface tension of people in the 18th century, like Young or Laplace, that cells adhere to one another, so they attract each other. And if you bring one cell to the surface, it has less neighbors, so it has less attractive interaction, so it costs energy. And that's what creates the surface tension of these aggregates. The order of magnitude is low compared to the surface tension of liquids. It's like 1 millinewton per meter. Now, the experiment that was done in Lyon by Mr. Rachbel here is that they took two of these spheroids and they pushed them against one another. So they force these two spheroids to merge. And what you see is that it does exactly like the very old experiments on liquids. The spheroid fused and makes this kind of peanut shape, which rounds itself because there is a surface tension. And this picture looks extremely much like experiments in the 19th century of scientists like Plateau, who are looking at fusions of droplets. So what this kind of suggests is that not only there is a tension, but there is also viscous flow in there. Now, if you look at the size of the neck here, for a viscous liquid now, you find that the square of the size increases. It increases, the square is proportional to the radius. It's proportional to the surface tension times the time divided by the viscosity. Now, here is a plot of the square of the radius as a function of the times the original radius, which is here. And you see that it's a beautiful straight line. And you can deduce from that the ratio of what's called the capillary velocity, which is gamma over eta. Now, if you measure the surface tension by other means, for instance, by squeezing the spheroids, you get a viscosity of tissues, which is very large. It's like 10 to the 5 Pascal seconds, which is 8 orders of magnitude larger than the viscosity of water. Okay, so I'll go back to that. What I was telling you is that this spheroid looks like droplets of cells with a viscosity and a surface tension. And for most purposes, you can describe them as these droplets of cells, except that they are growing. So they are growing because cells divide and because cells die. Now, if you write the conservation equation for the cells, so rho is the number of cells per unit volume, d rho dt plus the divergence of the flux of cells, where v is the local velocity, is not equal to 0. There is a creation term, so a rate of cell division times the density, and the rate of cell death, and cell death in this case is called apoptosis, times the density. So that's what creates new cells, and that's what destroys new cells. This rate of cell division, or the rate of cell death, depend on two things. It depends on the what I will call the biochemical states of the tissue. It depends on nutrients. It depends on the fact there is oxygen. It depends on what is called growth factors. So all of that is necessary for the cells to live and to divide. But it also depends on the mechanical state. So the mechanical state can be characterized either with the density, or if there is an equation of state in an equivalent way by the pressure of the tissue. Now, the pressure is something a little bit complicated for tissue. I have here this old drawing that we made in the first paper that we made on tissues, but I still don't know a better way to introduce the pressure. So this is kind of a thought experiment where we make a box, and the box is permeable to everything except for cells. We put a piston inside the box and a spring connecting the piston to one side, and put the cells on another side. When there are very few cells, they are very happy and they divide very fast. So they invade the space, they become confluent, and then they push the piston and compress the spring. Now, as I told you, the cell division decreases, the cell apoptosis rate increases, so cells die more, they divide less, and the growth rate, which is a difference, decreases. And at some point, you reach a force here due to the spring, where the cell division on average is equal to the cell rate, and you reach a steady state. So this steady state is what's called the homeostatic state. It's the state where the cell division is equal to the cell death. Cell division rate is equal to the cell death rate, and it's a property of the tissue. It's given by this equation that pH Kd is equal to Ka. Now, in order to illustrate that, I will show you a very simple experiment on which we worked a few years ago. So this is a spheroid that we put in solution. The spheroid grows, but we exert pressure on it. And the pressure is exerted by osmotic effect. They put polymer around the spheroid. And you look at the growth of one of these spheroids under external pressure. So they did it with many cell lines. I will only focus on this one, which is the one where the actual detailed measurement was made. It's a cancerous cell of mouse from the colon. The different lines here correspond to spheroids growing under different pressure. So this is a free spheroid without pressure. And you increase the pressure, so it grows more and more slowly, which is what I told you earlier. And if the pressure is too big, it stops to grow. If you go above something like 10 kilopascal, it stops to grow. If you go to longer time than that, I didn't insist on that, but if you want orders of magnitude, the cell division time is of order of 1 day. And to do experiments like this, you see here the first 10 days. And if you continue, there is a saturation that long time. It takes about 25 days to get saturation. There is a second experiment that they made is, so at a given pressure, you label the cells which are dividing and you label the cells that die. You make a cut and you can see where the cells which are dividing are, and the blue ones are the cells that divide. You see that the cells divide on the outside and they die inside. Now, if you have a steady state, it means that cells have a velocities that are created on the outside and they move inside and they die inside. The theory that we have for that is infinitely simple. We assume that the cells are incompressible. In this case, the number of cells is the same as the volume up to a constant density. So the volume variation has a bulk volume term, K division minus K apoptosis. And that's negative because you know that inside cells die. And then there's a surface term. So the surface is the volume to the 2/3, and they divide over a thickness lambda that you can measure, for instance, on this image. And then you can fit the curve. I didn't put a fit here, but the fit is almost perfect. And you can get two coefficients, this surface growth term here, and the negative growth rate in the bulk. The extra surface growth term is almost independent of pressure, so there is nothing much that you can get out of that. If you look at the growth rate in the bulb, which is negative, it's indeed negative, and it varies with pressure. If you increase pressure, cells divide less, they die more, so it should become more negative as you increase pressure. Now, if you go back, you see that at zero pressure, it's still negative, but you could extrapolate and you would get the pressure at which it vanishes, and this pressure is negative. Now, you might say this pressure has no meaning because it's negative. It would have no meaning if this were in an equilibrium system, but in a tissue, there is cell division and cell death, and negative pressures can be stable in a tissue because of cell division and cell death. Now, the last point is, why do cell divides mostly on the outside? There are two reasons. One is obvious, and the other one is a little bit less obvious. The obvious reason is that in order to divide, cell needs nutrients. The cells on the outside are more exposed to the nutrient. Nutrients diffuse in, they are eaten by the cells, and the cells inside have less nutrient. So the cells on the outside divide more. There's also a mechanical effect. When a cell divides, it needs to grow, and in order to grow, you have to push the neighbors. It's easier to push the neighbors on the surface because you have less neighbors. We made numerical simulations. In numerical simulation, you can turn off the nutrient effects, and we still see these effects that cell divides on the outside. So this is a very simple experiment. What we are doing now, and this is something that we do with Martin Benhamar and the student in collaboration with biologists at Institut Curie, Philippe Benarroch and Jovan Nikolic. These people are interested in immunological treatment of cancer. And they are interested in some cells of the immune system, which are called macrophages. And macrophages are very often present in tumors. And if you have a tumor and they find macrophages in there, it's a bad prediction, which means that macrophages, when they are there, promote tumor growth. What Jovan Nikolic and Philippe Benarroch do is that they make spheroids that I showed you before, but they make spheroids with two types of cells now, the cancer cells and the macrophages. So I will show you three types of spheroids. The ones with only the cancer cells, which look like the previous one. You see the time scale here is days again. And here we are almost on the steady state for the size. There is an intermediate cell, which is not quite a macrophage. It's a cell that hasn't differentiated into a macrophage. It's called a monocyte. They mixed monocytes and cancer cells. And you see a spheroid, which is almost the same as in the absence of monocytes. What you see around here is dead cells, which are floating at the bottom of the experiments. And they do the experiments with macrophages. And here it's pretty different. You see that there are many, many small spheroids, and that these small spheroids aggregate to make a larger macrophage. Now, if you measure the total number of stem cells, compare the pure cancer cells and the cells with macrophages, you indeed find that the proliferation is larger in the presence of macrophages, and that there are more cancer cells if you put macrophages. Now, there are two reasons for that. One reason is that there might be a direct effect, chemical effect on the macrophages on the cancer cells, that they just increase the cell division rate. But there is also a physical reason. You see that here, there is you make much more macrophages and you make many of them. What this means is that the macrophages favor the nucleations of the spheroid, and it makes smaller spheroids. But if the spheroids are smaller, most of the cells are exposed to the nutrients, and they divide more because they have more nutrients. So one reason would be that because macrophages favor cell division, favor nucleation, the surface of all the spheroids is larger, so nutrients are more efficient and cells divide more. So what they are doing now is that they want to have the structure of these spheroids where you have macrophages and cancer cells. So we try to model that. And the idea that we had is that maybe these two types of cells phase separate into the spheroids. And so what we wanted to do is some kind of canhilliard theory for phase separation combined to the growth of the spheroid. So I won't go into the details of that, but we use an effective free energy for a three-component system. So the three components are the two cell types and also the fluid which is inside in between the cells here, the intercellular fluid. The type of free energy that we use, because we know about polymers and systems like this, is a Flory-like free energy. We include cell proliferation, so we include cell division and cell death, and that comes into the conservation laws, the same conservation laws I showed you before. And what we use is the canhilliard theory for the kinetics of phase separation for this three-component system. Now, we haven't done it with macrophages yet because they don't have the experimental results, and they're trying to measure the effective interactions between two macrophages and the effective interaction between a macrophage and the cancer cells. So what the student Joseph Ackermann did, he solved these canhilliard equations numerically, including the growth of the system, for a system of two cells. So there are the blue cells and the red cells here. The blue cells divide, but when they divide, they make a blue cell and a red cell. And he puts interaction parameters in such a way that the two cells phase separate. So in the beginning, we have very few red cells. The blue cells divide, so the number of red cells increases. The red cells move to the surface, and after some time, you have this kind of strange structure where you have aggregates of red cells at the surface of the spheroids, and these aggregates grow slowly, and we haven't been able to go up there now, but what we think is that in the end, you would have a layer of red cells around the blue cells. Now, the reason why he did that is that there were experiments did by Chinese people that were exactly in this configuration, and these pictures that we obtained numerically exactly reproduce the result of this Chinese team. So that's this kind of thing that we're doing now. The output of this for me is that these multicellular spheroids behave as liquids. They have a surface tension, they have a viscosity, they grow, and also they can phase separate, and they phase separate exactly like fluid, and you can more or less describe the phase separation by canhilliard theory, or kind of a non-equilibrium version of canhilliard theory. What I want to do now is kind of go against that and show you that tissues are not always liquids. Sometimes tissues behave as solids. So this I will do with another system, which has nothing to do with cancer cells. These are what are called human-induced pluripotent stem cells. And this is a collaboration that we have again with Martin Benhamar and Joseph Ackermann, with experimentalists and biologists in Bordeaux. So these pluripotent stem cells are stem cells that can differentiate into any cells that you would need. So they are pluripotent in the sense that they can differentiate into anything. And they are used for cell therapies, which means you inject those and they will differentiate in the cells which are needed in the tissue while you inject them. In order to do that, you need many of them. And the strategy is to make aggregates of these cells that will grow and produce these stem cells. However, you need that when they grow, they remain stem cells. Now, what people were doing is they were trying to make spheroids, as the one I showed you, and these didn't work. It didn't work because they were producing many cells, but the cells didn't have the stem cell character and couldn't differentiate anymore. What Pierre Nassois and Philippe Cohen did is they found conditions where the cells didn't make spheroids, they make what are called cysts. So a cyst is a spherical shell of cells which is hollow. So the cells are here, And at this stage, which I'm showing, the cells span from this external surface to the internal surface. They form a sphere. They form a spherical shell, and inside you have fluid, and the fluid goes freely between in and out. So the assumption that we have is that the pressure is the same inside and outside. So what they did study is the growth of these cysts inside a polymer capsule that the one that I showed you before. So I have several experimental results to show you here. So this is a very small cyst. The dark part in the center is the fluid. You see the shell of cells around, and there's a dark region around, which is a lumen of the polymer capsule. This cyst grows, and at some point it touches the capsule. It wants to grow and pushes against the capsule. Now, another way to represent that, which is not so easy for me to read, but that biologists love, is to make what they call a chymograph. So they cut along one line here, and this coordinate here is the position along the line over which they cut. So if you cut here, there is the alginate capsule, which is at the bottom here. There is an alginate capsule at the top. You see the layer of cell, this one at the top here, and you see the layer of cell at the bottom here. We characterize that by two radii. There is the inner radius here, which is this one, and there is the outer radius. Until this line here, the cyst doesn't touch the capsule, so it's free growth. And during free growth, the inner radius and the outer radius both grow. At this point here, the cyst touches the elastic layer outside. This is the lumen, what's inside, and you see that the inner radius almost stops growing. The outer radius grows strongly after that. So at this point, the cyst doesn't grow freely. It pushes against the elastic medium, which is a capsule. We also look at a picture that occurs at long times here, and you see that the form of the internal surface is very unstable. And that, to me, but mostly to Martin Benhamar, who is an expert on solid mechanics, looks like a bioinstability. If you compress an elastic medium, it buckles. So we believe that this is a buckling of this cell layer, which is here. And if it's the buckling of the cell layer, which is here, it has to be an elastic behavior of this cell layer. Now, what we did is we treated this cell layer using what people call a morphoelastic model. So this is kind of a complicated word to say that we want to take into account both elasticity and growth. So the way you do that is you, as in any case in solid mechanics, you consider a reference state, and I call the reference state with capital letters. dr here would be the length of a small element. This small element grows and is deformed. So the elongation tensor, which means the tensor that deforms it to the real state, is a product of two tensors, a growth tensor and an elastic tensor. And the order of the two tensors is important here. It first has to grow and then to be deformed. Now, if dr is the real length of the element, this big R is the reference state. Gamma R is the growth in the R direction. So this is the reference state when it has grown, and this is the elastic deformation. Similarly, in the theta direction, the reference state is R times the growth times d theta, and the actual state is small r times d theta. So this is the elasticity in the theta direction. As I did before, I will assume here that the tissue is incompressible. So the square of alpha theta times alpha r is equal to 1. And the determinant of this growth matrix here is just gamma r times gamma theta squared. So this is how we define our system. And then we discretize time And we assume that the growth occurs step by step, that there is a growth rate that I call kappa, K here. There's a growth rate in the R direction. There is a growth rate in the theta direction. And these growth factors, gamma, are just the logarithmic, the rate. The growth rate is a logarithmic derivative of the growth factors. Now, as I did for the spheroid before, I will assume that the growth rates, K, depend on stress, and we assume that it depends linearly in the Cauchy stress. Now, you did an elastic model. We take the simplest nonlinear elastic model that exists, which is the neo-Hookean model. So it only has one modulus, which is a shear modulus, and this is alpha r squared plus alpha theta squared. So we calculated the stress from that. You write force balance, assuming that the pressure is the same on both sides. If there is no, if the cyst doesn't touch the elastic capsule, and if there is an elastic capsule, the cyst pushes on the elastic capsule and the stress is continuous. So we solve these equations. The experimental points are the green points here. This is the outer radius of the cyst. This is the inner radius of the cyst. And the line is a morphoelastic model. The parameter that we fit is the elastic modulus of the cells. There is no confluence until this time here, so you have to wait 7 days or so before you touch the capsule. Here, the deformation is small, which means the stresses are very small when you are in a cyst. They become much larger when you when you touch the capsule. They become much larger and the cell layer becomes very thick. Now, if you go back to the previous picture, there's an image that I didn't comment. This is a test that the cells remain stem cells. They should express a few genes, and these are the genes which are tested for the cells to remain a stem cell, and in the cyst, they remain a stem cell. So the conclusion is when the cells grow, when the cysts grow, elastic stresses are generated. If these stresses are small, the cells remain a stem cell. If the stresses are large, the stresses are too large, and the cell don't remain a stem cell. So the strategy to make many stem cells would be to make cysts and to grow them not too long until they don't touch the capsule where they are. Here is another representation of the same thing. This is the outer radius as a function of the inner radius. They grow the same way because you have a free sphere when the cell don't touch the capsule. And after what we call confluence, the outer radius grows strongly and the inner radius doesn't grow. So the curve goes up here. So what I want to retain from that is that in certain cases, tissue behave as soft solids. The elastic modulus is something like 10 to the 4 or 10 to the 5 pascals for the cell. So it's of the same order of magnitude as the elastic capsule. So cells can be liquid, cells can be solid, and of course they can be viscoelastic. So in a sense, the way I view tissues here is that they are viscoelastic fluids. Sometimes the mechanical relaxation rate is small, and it's always a liquid. Sometimes it's large, it's always a solid. In many cases, the relaxation rate is fixed by the cell division time. Now, this is not the end of the story, because in some cases, you have to go beyond that and consider the cells as active liquids. How much time do I have, Rodrigo?
Let's say 10 minutes more.
Okay, so I will show you that. So these are monolayers of cells, and these cells are elongated. What you see here is an image of these elongated cells. The surface is treated, and it reveals the orientation of the cells, and you see that the cells are parallel. So what this tells you is that the cells are in a nematic phase. How do I know that they are in a nematic phase? I know because there are defects in the order here, and this defect is what's called a -1/2 defect. It's sketched here. It has this 3-4 symmetry. If you turn around the defect, the orientation turns by minus pi. This defect here is called a +1/2 defect. The orientation is sketched here. If you turn around the defects, the orientation turns by plus pi. And if you had a polar system, which means if the cells weren't only parallel, but they were all oriented in the same direction, if they had an arrow on the head and the tail, you wouldn't have this +1/2 and -1/2 defects. You will have +1 defects. So the fact that you observe these defects tells you that this is a nematic phase. Now, what the team of Pascal Silberzahn did is not only did they measure the order of the cells, but they also measure the local velocity field. Maybe the most spectacular stuff of this experiment, so there is a velocity field, is to look at the scale here. So the scale bar, this is 1 micron per hour. So before you can do that, you have to wait for days. But after you wait for days, you can measure velocity field. And you see that the velocity is strong at this place, which is this defect, and is strong at this place, which is also a +1/2 defect. But at this one, nothing happens. Now, this is kind of a flashy picture to show the same defects as these ones. What this is telling you is that +1/2 defect move and -1/2 defects don't move. Tells you two things. First of all, that this is an active process. Cells need energy to move. So there's no way you're going to make a theory of that with a passive theory. And the other thing is kind of see, this one gives you the orientation of the cells. And for me, the easy one to see is this one. You see a defect, a +1/2 defect that moves in the direction of the arrow. And what Benoît Ladoux figured out is that cells don't die in this layer. They are extruded from the layer, and they are extruded in the ahead of the defect. So the defect is here. The cell is extruded here, so it would be extruded a little bit outside of the defect, as shown in this picture here. Pascal Silberzahn did experiment in the same direction. So you have the +1/2 defect here. What they observe is that on top, in this tail region of the defect, the monolayer doesn't remain a monolayer. The cells are extruded, and they form a second layer. However, these two experiments are not exactly the same, because in this case, cell extruded in the head of the defect, and here cells are extruded in the tail of the defect. Now, in the 5 minutes that I have left, I will explain to you very briefly how we take into account the activity. This is a nematic system, so the orientation of the cell has a direction that I call P, and there was something which is invariant when I change P in -P. So I need a tensor, which is this nematic tensor, P alpha, P beta, -1/2 of delta alpha beta. There is an energy scale in the cell. Energy in the cell comes from the hydrolysis of a molecule called ATP. And this hydrolysis provides for each molecule hydrolyzed an energy, which I call delta mu here, which is of order 25 kT. Now, if you have a tensor here, you can make a stress tensor proportional to this active stress. So it will be Q, the nematic tensor, and times some constant. Now, this stress doesn't exist in equilibrium nematic, so it has to be some non-equilibrium stress, and it's proportional to delta mu. Delta mu is positive, and then there's a prefactor. For some reason, in the beginning, we put a minus sign. The point I want to insist on is that this is not a pressure. This is kind of a normal stress difference. And this is what I try to explain in this small sketch here. This is the orientation of P. And there are two cases. Either the stress extends in the orientation of P and compresses in the perpendicular direction. In this case, the tissue is contractile because if you cut it from its environment, so you suppress the stress that pulls it outwards, it will contract itself along its orientation. And in this case, the stress is positive, so zeta, which is a prefactor here, is negative. For some cells, it's in the other direction. So along the polarization, the stress is negative, and the tissue tends to extend because you need to compress the system to make it stable. So the output of that is that in non-equilibrium active nematic, you have a stress proportional to the nematic tensor. There is a strong implication of that, which is that if there is a gradient of orientation, there is a stress associated with a gradient of orientation. So anytime you have a non-uniform orientation, there is a local stress on it. So let's go back 1 minute to defects. So this is the positive defects. I won't talk about the negative -1/2 defects here. Of course, in the defect, there is a gradient of orientation. So there is a local stress. And what you can do is calculate the divergence of this active stress, and that gives you the local force. Now, if the tissue is contractile, tends to contract, the active force, the divergence of the stress, goes along the direction of the defect, and it points toward the tail. So it points toward the tail. It means it pushes the defect in the direction of the tail, and the defect moves in this direction. If the tissue is extensile, you can calculate the active force. It's parallel to the defect again, but this time it pushes towards the head. So if the tissue is extensile, it moves towards its head. If the tissue is contractile, it moves towards its tail. And then you can calculate using almost classical hydrodynamics, that is active hydrodynamics. You can calculate the cell velocity field. It's sketched here, so the defect orientation is here. The velocity lines are these flow lines, are these blue lines. There is a vortex here that turns like this. There is a vortex here, and the tissue is pushed towards the right if the tissue is contractile, is pushed towards the left if the tissue is extensile. Now we calculated the velocity of this tissue is proportional to the active stress, and what makes the friction is the viscosity of the cells times the friction of the substrate, and the denominator is just the square root of the two. Now, I will make a long story short. You can also do it in the presence of cell division. And in the presence of cell division, the growth rate, the proliferation rate depend on pressure. So division rate minus death rate depend on pressure. It vanishes at what I call the homeostatic state. This is a strange thing because even for an incompressible tissue, the divergence of the velocity is non-zero. So this is hydrodynamic, but with something which is very non-usual here. And from this, you can calculate the pressure map, which is plotted here. The tail of the defect is on this side. The head of the defect is on this side. We calculate the divergence of the velocity. It's negative here, so the pressure is positive. The divergence is positive here, so the pressure is negative. So cells are extruded at the places where the pressure is positive, so they should be extruded on this side. If the tissue is contractile, if the tissue is extensile, they are extruded on the other side. So this seems to explain the system. However, this is not the whole story, and the velocity field doesn't fit at all with the experiments. So we have a qualitative explanation, which is associated to the fact that cell division and cell rate depend on pressure. Maybe the extrusion is not controlled by the cell division rate, but at least this is what we are on this system. I will skip that, which is what happens if you go to very high activity where there is some kind of active turbulence. and which we are working on. I won't talk about it. If I try to summarize, what I try to tell you is that tissue can behave as nematic viscoelastic fluid, but they are growing. There is a coupling between the mechanics and the growth. The growth rate and division rate and death rate depend on the local pressure. There is a viscoelastic relaxation time. We have a model that relates this viscoelastic relaxation time to cell division time. So the viscoelastic relaxation time in many cases is of order of days. And what I showed you here is that there are elastic instabilities. Now, in the beginning, I showed you this structure of the intestine, which is this array of protrusions, which makes a square lattice. We have a model for that, which is also based on the buckling instability of the intestine. So I think there is very original physics to do with tissues. The other point that I want to make is that you can bring that back now to real cases of tissues. I talked about tumor growth. I talked about Drosophila embryos during development, and I talked about cell therapies. There is one thing which is missing here, is a coupling between mechanics and growth and signalization, so chemistry. This is something which is obviously necessary, and some people are working on that as well. I will stop here. Thank you for your attention and answer your questions. Thank you very much.
Thank you, Francois. So we have time for questions. I think that there was a question by Marcel, but I lost it because when we change the presentation, the Zoom session, I don't know if Marcel is still there with the question. No. Yes, I have one question. Just when we change in the Zoom session, that you were saying that when these two droplets of cells merge, the viscosity was very high.
Yes.
Is there an explanation of this high viscosity?
If you assume that the tissue is a viscoelastic material, the viscosity is the modulus times the viscoelastic relaxation time, which is called Maxwell relation. The viscosity of tissues is something like between 1 kilopascal. The elastic modulus is between 1 and 10 kilopascal. The relaxation time is 1 day. So if you multiply that, you get something which is very large, 10 to the 7 Pascal seconds or 10 to the 8 Pascal seconds, which is 10 orders of magnitude more than water.
Okay, but in another time scale, the viscosity should be closer to water, or?
So if you look at tissues at small time scales, you push on them, they are elastic because they are way below the viscoelastic relaxation time. So they are viscous, but they are viscous if you look at extremely long time, days.
Okay. Yes. I have the question of Marcel that he was asking if this vertex model can be used in non-flat tissues.
If the what?
The vertex model.
The vertex model. Yes, it can. So at least the way I did it is we used the vertex. We made a three-dimensional vertex model. So in the picture I showed you, I draw the cells as cylinders. Now, if you assume that the surfaces at the top and the surface at the bottom are different, you make kind of conical shapes, and that creates bending of the layer. And then if you bend the layer, you can have spherical shapes, you can have cylindrical shapes, or even more complex shapes. For instance, my last point on cysts, you could describe cysts with a vertex model. They form naturally in the vertex model. Okay. Or you could imagine more complicated shape. I mean, you could imagine surfaces with a negative curvature and see what happens. I don't know any work with complicated shapes, but I know works with people doing calculation with a vertex model with cysts, for instance.
I think that Aneso has done some work with negative curvature. So okay. Please. Okay, there's I don't know if there's another question. I have one from the chat as well. That Maria Luisa asked that she noticed that the line, and in the first part, the line tension can be negative. What is the difference between positive and negative line tension?
What is the difference between positive and negative line tension? Yes, so this is a very good question. So in practice, what creates if you have two cells, of course, the cells are three-dimensional. So they have two top and bottom surface, but they have lateral surfaces. And what creates a line tension is the interaction between the lateral surfaces. And there are two contributions. There is a contribution from the cortical region of the cell itself, and that's a positive contribution because the cortex is contracting in a cell. But there is a strong contribution from the adhesion between cells, and this one is negative. So if the adhesion dominates, the line tension can be negative. And in principle, you could tune that by modifying the contractility of the cells.
I think that Pablo de Castro has a question. Can you raise your microphone or?
Yes.
Yes.
Let me make the question myself. Yeah, so have you seen slow phase separation kinetics in the spheroid canheliac model? Because, for instance, a cell could take a long time to reach its final droplet.
So of course, this is also something we studied. First of all, if you take before the phase separation, you could look at diffusion of cells inside the spheroid, and that's extremely slow.
Yeah.
So we see phase separation kinetics, at least in the numerical calculation that I show, and the phase separation is very slow, if only because the time scales are very slow because the tissue is very viscous.
Yes. Great. Thank you.
Okay, there's time for one more question. Well, I have one that you briefly said at the beginning that there's some difficulties on building these macroscopic or hydrodynamic theories for active matter. Can you comment on that?
So there are two things you can do. Either you build what I kind of sketch here for active liquids, which means you use only the symmetries of the problem. Which means if I'm looking for a stress, it has to be proportional to the tensor that you have, which is a nematic tensor. And there is a systematic way to do that. You use the approach that has been created for the hydrodynamics of liquid crystal. And this is what we did for the theory of active materials. And this is the hydrodynamics that I use. The other thing you could do is that you could start from a more macroscopic model and try to coarse grain it little by little to reach the macroscopic scale. So starting from this vertex model, you could wish to make a continuum theory. People haven't done that completely. The only thing I know is, how should I say that? The kinematics has been done. In a sense, people have defined properly velocity gradients and things like this inside the tissue. They haven't related it to forces. And that's kind of what we are doing. Even the strain tensor is not obvious to define. Because, I mean, at short time it's elastic, but you need a reference state to define elasticity. And the cells are dividing, so it's not even easy to follow single cells. So you have to be a little bit clever to define the strain tensor.
Yes, now I see. Yeah. Okay, so it seems that there's no more questions. So I would like to thank you, Francois, for staying until very late at night there.
Okay, don't worry.
And thank you very much. Thank you for everybody. And as always, the talk will be uploaded to the YouTube channel and the Facebook channel of the physics department. Okay, so thank you, everybody. I'm going to stop recording.
Okay.
See you next week.